So the IMF, through its Global Myeloma Action Network Advocacy Initiative, known as GMAN, is pleased to present the first of 2 webinars this year geared for both our GMAN members and the larger global audience. For those of you not familiar with GMAN, it is a group of 37 myeloma organizations from around the world with members in North and South America, Asia, Oceania, and Europe. The objective of GMAN is a simple one. It's to improve the lives of myeloma patients around the world. But that objective is complicated by differences in cultures, governments, healthcare systems, and economics. GMAN hopes to be the bridge between those gaps through collaborative efforts among members. Very shortly in June, many GMAN members will be meeting in Copenhagen, Denmark for our 4th annual summit, where we will address such things as disparities in access to treatments, global awareness of myeloma, and patients' rights. For more information on GMAN, please visit our website at gman.myeloma.org, or you can contact myself, Ray Wiesek, and there's my information up there on the screen, rwiesek@myeloma.org, or you can also contact my boss, Megan Busby. She's the Senior Director of Advocacy at mbusby@myeloma.org. I also just wanted to show real quick, here are some of the members of GMAN, and many of them will be at our meeting in Copenhagen, Denmark this June during EHA. This webinar is focused on the recently approved myeloma treatments here in the U.S. by the FDA that are now being approved in other parts of the world. With us this morning is Dr. Rafat Abonour, Professor of Medicine, Pathology, and Laboratory Medicine at Indiana University, where he's worked since 1995. Dr. Abonour established and directs the multiple myeloma and plasma cell program at Indiana University. He is principal or co-investigator of several completed and ongoing clinical trials. Dr. Abonour has authored or co-authored numerous peer-reviewed manuscripts, book chapters, and review articles. His work has been published in key medical research journals, including Blood, New England Journal of Medicine, and Nature Medicine. Needless to say, we're very lucky and pleased to have Dr. Abonour with us this morning. And so without further ado, I will turn this presentation over to the good doctor. Dr. Abonour?
Thank you very much for your kind introduction. Good morning and good afternoon, everybody. And it's a pleasure to talk to you about this exciting time in the multiple myeloma battle toward curing patients. First of all, I just want to show you that each decade we are making progress. On the next slide, you can see that the overall survival of patient with multiple myeloma is continuing to get better. So with each decade, we having more patients surviving multiple myeloma. I think the reason for that is 2 things. The first one is that we achieving better responses. We're getting more patient into complete remissions, and we maintaining the complete remission. So that is really the key point that I'm trying to drive today, is that we need to eradicate myeloma early and achieve a complete remission early. That will translate, hopefully, to 100% survival of all newly diagnosed patients with multiple myeloma. So what's the story in the last year? On the next slide, you can see that we have, plethora of drugs been approved. It's unprecedented. 4 drugs, the Penovistat, Daratumumab, Activism, and Elotuzumab. And obviously, the CAR T cells not exactly approved, but it's getting a lot of publicity. I think in U.S. We have this magazine called Parade last Sunday. So every patient I saw this week brought this copy of it to ask me about it. So I thought I'll mention it a little bit here today. So on the next slide, we're going to go through these items one at a time. First is the Penovistat story. So Penovistat is an HDAC inhibitor that when it combined with bortezomib lead to a better blockage of this proteasome, and then by doing so you get more cells undergo apoptosis or cell death. So what were the result of such approach? Basically, in the next slide, the panorama study design was to address whether by adding Penovistat to Velcade dexamethasone, can you improve the response rate and delay progression? And these were patients who never received bortezomib or Velcade before, or if they received it, they were responded to it. So the two arms were Velcade Dex, and the second arm at the top was the Pinovistat, 20 milligram giving on day 1, 3, 5, 8, 10, and 12, with the Velcade, the classic dosing, day 1, 4, 8, and 11. So I'll show you the result of this trial on the next slide. Basically, the progression-free survival improved by 4 months. The median was 12 months when Pinovistat was added, and the Velcade arm was only 8 months. The number of patients responding was higher, also 60% versus 54%, and there were more patients achieving a near complete remission, 27 versus 15%. So it looks like it's a very good regimen with improvement in the response, regardless if they had multiple line of therapy or not. The major issue that was the adverse event in this trial, as you can see, there was significant diarrhea seen in 25% of the patient compared to the control arm, which was only 7%. Neuropathy was similar, did not increase by adding the Pinovistat. So I think it's an interesting combination with promise. I'll show you on the next slide. There are a plethora now of HDAC inhibitor that are being tested, and they are different. One of them, for example, the Clonostat, it has probably more HDAC activity inhibitions, and you can see here in this phase 1 study, there were 25% response rate in heavily pretreated patient, and it can be combined with lenalidomide. There's also a combination of Pinovistat with carfilzomib. As you can see in this phase 1 study, the overall response rate was 77%. There were 1 patient achieved a complete remission, 10 patients achieved a very good partial response. So I think the HDAC inhibition is going to be part of the game here in multiple myeloma. There will be several competitor of Pinovistat on the market, and the question which one will be the best with the least side effect, and that's what we do the study for. On the next slide, I'm going to talk about Activism. Activism was approved in the U.S. in November, which is an oral proteasome inhibitor. And the pivotal trial that led to the approval of the drug was this trial. We established that Revlimid dexamethasone is an appropriate treatment for relapsed patient. So can we improve on that by adding Activism? So this was a large study of 722 patients assigned to either Activism, lenalidomide dexamethasone, or lenalidomide dexamethasone. And these are the nice thing about Activism, it's a pill, again, giving on day 1, 8, and 15, 3 tablets a month, instead of going to the clinic to have an injection or IV form of the proteasome inhibitions. So what were the results of this trial? On the next slide, you can see that there is, again, delay in progression free survival from 14.7 months with Revdex to 20.6 months with the Activism Revlimid Dex. The overall response rate was also better, 78 versus 71%. Very good partial response was also better, 48 versus 39%. So clearly improve the response rate, the depth of the response, and delayed progression. In terms of adverse events, that was a very pleasant surprise. Number one is that the incidence of significant diarrhea was quite low, 6%, and the incidence of significant neuropathy was also low at 2%. And the benefit of this combination was established even in high-risk cytogenetics. So it's an interesting regimen, all oral regimen with excellent response rate, period. So that's good. So let's move on to the next slide and talk about the 2 monoclonal antibodies that has been approved So the first one is, for example, what is monoclonal antibody-based targeting multiple myeloma? So the way it works is that the antibodies can basically bring the myeloma cells to what we call them effector cells, the cells that can sort of attack the myeloma cells and kills it. So this is called antibody-dependent cellular cytotoxicity, or it can engage the complement system, such as C1q, as you can see in the middle. So daratumumab does that, or it can lead to direct myeloma cell kills by targeting what we call apoptosis pathway. So you can see there's a lot of different antibodies on the market, from CD40 to SLAM7 to CD38, and you can see that daratumumab works in different areas, from directed cellular dependent toxicity, complement mediated, or direct apoptosis. Elotuzumab does work by bringing the effector cells to the myeloma cells, and that's probably why it's so important to understand that, because I think that will explain the difference between the two monoclonal antibody. Daratumumab probably as a single agent has more activity than elotuzumab. So on the next slide, we're just going to show you the two targets that are being studied now, the CD38 and the SLAMF7. So what is daratumumab on the next slide? Daratumumab is a human manufactured immunoglobulin IgG that targets CD38. And I think that's important to remember that it's an IgG and it's a monoclonal antibody, so it will be seen on serum protein electrophoresis, and you need to let know the blood bank that the patient is getting daratumumab, because it can interfere with typing of for blood transfusion, and also need to let the lab know because it can be misread as the persistent myeloma, but actually it's the presence of this monoclonal protein in the serum. So anyway, so daratumumab attacked CD38 cells and caused their death. So let me show you the results of, on the next slide, of single agent activity of daratumumab. So these are patients who are heavily pretreated. They have seen all the available proteasome inhibitor and immunomodulatory drugs. And what you see here, as a single agent, the overall response rate was 31%. So really a very impressive single agent activity. And there were some patients who achieved the stringent complete remission and complete remission. There were 10% of the patients achieved very good partial response, and partial response in 18%. So outstanding single agent activity. The next slide is to show you that when you respond, you can maintain and improve your progression-free survival. The patient who, the responder are on top, and the patient who were progressing while they're getting the treatment didn't do well. So it's important to see responses for that drug to work. On the next slide, showing you that also the overall survival is influenced by the response to the monoclonal antibody daratumumab. So if you were responding, you're going to live longer than the patient who have stable disease or minimal response. And obviously the patient who were progressing didn't do as well. So very encouraging single agent activity that is now leading to a combination therapy. So on the next slide, you can see that daratumumab in combination with bortezomib and pomalidomide is undergoing investigation. And you can see, for example, when it's combined with Velcade dexamethasone in a newly diagnosed patient, the response rate was 100%. When it's combined with bortezomib, thalidomide dexamethasone, again, the response rate was 100%. So even if you're not doing transplant and you combine it with Velcade, melphalan, prednisone, you're getting 100% response rate. So, and in a patient who have failed 2 lines of therapy, we're getting improvement in the response rate to 50% when it's combined with pomalidomide. So clearly a very important drug, and it renders itself to combination therapy because of the lack of overlapping toxicity. And that's going to be really a key and important drug even in the newly diagnosed patients. So on the next slide, we're going to talk about Elotuzumab, which is also a humanized monoclonal antibody that target SLAM F7, and basically the way it works by sort of bringing the natural killer cells in closer to the myeloma cells and cause the cells to die. So again, it doesn't work alone by itself, does not cause apoptosis by itself, but does work by enhancing the sort of innate immunity of the patients to try to eradicate the myeloma cells. So what happened? On the next slide, Elotuzumab was studied as a single agent in 35 patients, and there was dose titration from half to 20 milligram per kilogram, and there was stable disease in 27% of the patient. But when combined with Revlimid and Dex, the response rates jump up to 80%. For that reason, on the next slide, you're going to see the Eloquent 2 study that led to the approval of the drug in the U.S. And basically what you see here is that, again, we know that Revlimid Dex or lenalidomide Dex is the standard of care for relapse patient. The question is, can we improve on the response rate by adding Elotuzumab? Again, so you can see the sort of design is similar to that of the Activism study. So what did we find in this study when we take patients who relapse after 1 to 3 cycles of myeloma therapy? On the next slide, you're going to see the following. Number 1, is progression-free survival again improved? When you add Elo, its median is 19.4 months. When it's just Revendex, it's 14.9 months. The overall response rate is even better, 79 versus 66%. There were more patients getting a very good partial response, 33 versus 28%. And you can see the adverse side effects were really, really minimal advance. So it is a interesting combination that is look very promising also in terms of improving the response rate when it combined with Revlimid Dex, and the approval is in combination since single agent activity is very limited. So on the next slide, I'm going to show you that the patient who received Elo did better in terms of overall progression-free survival and response rate. So you can see on the top there's improvement in the progression-free survival, and there were also improvement in the overall response rate. So clearly a very powerful combination that should be further investigated and maybe early on in the course of the disease. Finally, I'm going to touch on the CAR T-cell immune therapy on the next slide. So the problem is immune therapy, slide please, is yeah, previous slide please. Thank you. So on this slide is really what happened is that, myeloma cells are very good at hiding from the immune system, and the immune system in myeloma patients is kind of lousy and not able to recognize the cancer cells. So some of the technology now is try to get the T cells to express a protein that can recognize the myeloma cells, and also a protein that will kill the myeloma cells. So that's really the principle of CAR T cells, is take the patient on the next slide, please. So what you're taking is you're taking the peripheral blood lymphocytes, and you take these lymphocytes and you, in the lab, put two genes, one to recognize myeloma and one to trigger and kill the myeloma cells. So that's dual gene transfer in the lab. So this is a very elaborate and complicated process. And then you infuse these cells to the patient and see what happens. So let's go to the next slide. So now the question is, what is the target that we need to sort of attack on the myeloma cells? And I think that's the problem with the early CAR T cells in myeloma, that the target that was used is CD19. CD19 is a B-cell antigen that can be present on lymphocytes and probably more common on chronic lymphocytic leukemia and acute lymphoblastic leukemia, but not much on myeloma cells. So that's why the group in Pennsylvania now start looking at a different antigen called B-cell maturation antigens, or BCMA, and they are doing the study with that. So what did they do? Then the next slide, please. So what they took is they took patients who have been refractory, relapsed, and what they did is they give them a cytoreduction to get rid of large bulk of the myeloma cell by giving cyclophosphamide, and also getting rid of some of the immune system by giving fludarabine. And after they did that, they give them the infusion of these CAR cells at a titrated dose from 0.3 million to 9 million cells per kilogram. So on the next slide, you will see the result. And basically, one patient achieved a stringent complete response, one patient achieved a very good partial response, and two patients achieved a partial response. The majority of the patients had a stable disease, 8 patients. So it is interesting. The problem with these infusions that sometimes are associated with what is called cytokines release syndrome. So the patient become quite sick, they drop their blood pressure, hypotensive, they end up in the intensive care unit on pressor for a while. So the issue is, what is that process? How can we alleviate that process, and can we maintain the response without the cytokine release syndrome? So I think it's need to be monitored, this cellular therapy. The alternative is going to be perhaps the checkpoint inhibitor, where you give drugs that will awaken the immune system and also make the myeloma cells visible to the immune system. So on my last slide, I'm going to say that really it's a great time for myeloma patients. We have several new options available to them at relapse. The design have been mostly to delay progression. I think it's really important to try to achieve a complete remission, because if we do so and we achieve a minimal residual disease, I think we're going to start curing some patients. And the longer the myeloma is sitting around and eradicated, the more likely we'll acquire genetic abnormality that will make it very difficult to eradicate. So I'm excited about where we are today, and I'm excited about where we will be tomorrow. So I'll be happy to take any questions.
All right. Thank you very, very much, Dr. Abonour. So at this point, we have a couple of questions that have come in during the webinar. The first was, with these clinical trials that you were discussing, are these for patients who had previous treatments, or are these frontline patients?
So most of the trials that I presented were for a newly diagnosed, for relapsed patient, and that's why these drugs are approved. But I showed you also an example of trials that ongoing, for example, with daratumumab in combination in the newly diagnosed patient. There are a lot of trials now, many trials, that are looking at these drugs in the newly diagnosed patients.
Okay, thank you very much.
You need to stay tuned for those results. I'm sorry. Some of them will be presented at ASCO in a couple of weeks, and some of them will be presented at ASH in December in San Diego.
Okay, fantastic. I have another question. Why was panobinostat approved with Velcade but not with Revlimid?
Well, I mean, I think if you look at the mechanism of action, you know, it does enhance the sort of blockage of proteasome, and so the combination, it's rational and scientific and led to the activity. When panobinostat used as a single agent activity, it's not that powerful. So the question, does it really add to Revlimid? Does it give synergy when you add it to Revlimid? And I'm not aware that's that's the case. So I think that's why panobinostat is being only tested with proteasome inhibition, such as carfilzomib, Kyprolis, and Velcade. Mm-hmm. And
okay,
I think it probably will be tested with Elotuzumab soon.
Fantastic. I have quite a few questions coming in, actually, so I'm going to try to keep these as short as possible.
Okay.
The next question I have is, one doctor tweeted that 11 of 12 patients have relapsed from the CAR-T BCMA trial. Is that true? Or do you know if it's true?
Well, I mean, I don't know the updates, you know, because they have not been presented, but I would not be surprised that these may not be lasting, because the question is that, can these T cells survive in the patients forever, you know, because these T cells are not going to regenerate themselves. They're not going to duplicate. And once these T cells sort of engage the targets, they may, you know, both of them will, you know, both cells will die. So it is, you know, it will be interesting whether we need to sort of, if this works well, that we need to keep injecting some of these T cells to eradicate the disease. So that's why I'm maybe more excited about the checkpoint inhibitor than the CAR T cells.
Okay, fantastic. Next question. For those who cannot tolerate Revlimid, what are the prospects for making Laurel available as a single agent?
Well, I mean, that's a very, very important question, and I, you know, to be honest with you, I do have the experience, you know, using Elotuzumab as a single agent, and I mean, with some low-dose dexamethasone, obviously, and it does work in some patients, and in some patients it doesn't work. The question is, you know, do we have the right dose? You know, the 4 milligrams, is it enough 3 times a week or not? So, unfortunately, the way the insurance company are now looking at this is that, you know, they don't want you in the U.S. to prescribe it without Revlimid, and you know, we're having a hard time getting it just as a single agent. But I usually fight for it, and I have seen activity without Revlimid. So I think it should be addressed one patient at a time, you know.
Right. I don't know if you would know the answer to this question since you primarily practice in the U.S., but are any of these newer agents or trials becoming available in South Africa?
You're correct. I don't know the answer to that. Yeah.
Sure. With DARA, is that a choice route for newly diagnosed patients?
Well, I mean, I think if a patient can afford it, I mean, I would probably include it in the newly diagnosed. As I said, there are many studies ongoing that will address the issue of the advantage of adding DARA to the newly diagnosed patient. I think rationally makes sense. It's a very good drug that can be combined with, for example, RVD, the Revlimid Velcade Dex, or Cybor-D, the Velcade Cytoxan Dex. It can be combined with those. And again, I am a believer today that it's so important to get complete remission as soon as possible, as safely as possible, because that can translate to curing patients. So I think, you know, it is being tested, and I think it may become part of the induction treatment.
Now, I have another interesting question. With all of these new drugs, is transplantation becoming a second choice?
Well, I mean, that's Very important question. So let me tell you something. I try to explain it to my patients, and I say, listen, when you have multiple myeloma in your bone marrow, the myeloma cells in the bone marrow, they don't all behave in the same way. Some of these myeloma cells can be active against one drug and then resistant to another drug. So it's like having a family. You have 5 children. Each child behaves differently. You have to address them differently. And the same thing happened inside multiple myeloma. And the scientific term for that is called clonal heterogeneity. We don't have the same myeloma cells all over our body. We have different myeloma cells behaving differently. So I think to be able to think that we can cure the patient with, drug A and B, but not drug C, it's probably not wise. I mean, I think it's going to require combination and sequence of regimen. So until we've proven otherwise, we know even in the setting of these drug Revlimid and Velcade and great results that transplant actually was better when added to these drugs than when it's delayed. So until we have evidence that we can eradicate myeloma cells, all of them, without transplant, I think we still should consider transplant, as an option and not a second option.
Okay, interesting. Here's another interesting one. Are other drugs for blood cancer being developed that may have activities for myeloma? If so, which ones and why are they being, or where and why are they being studied?
Other drugs being developed for, well, I mean, there's, if you look at the number of clinical trials ongoing in multiple myeloma, there's at least, I think, last time I checked, 199 phase 1, and there's about 250 phase 2 studies with different new drugs in patients with multiple myeloma. So there is a plethora of drugs, and companies are interested in studying multiple myeloma, and these drugs can work differently. So, for example, some of them, works on a certain pathway, ATK, or certain kinases inside the myeloma cells. Some of them are working on the immune system, so many drugs now have these called checkpoint inhibitor that they are testing in multiple myeloma. So plenty of stuff. I mean, if you go on clinicaltrial.gov, clinicaltrials.gov, and enter multiple myeloma and do the search, you'll find all these different trials that are being conducted.
Great, and that is a great resource for those that are not aware of it. And this will be the last question. Unfortunately, I want to be respectful of Dr. Abonour's time and let him get back to what he is doing in Indiana. So this will be the last question, and if anybody else has any others, they can email me and I will try to get them answered. So the last question is, in the CAR T therapy, one of our listeners thought the exciting benefits was that those engineered T cells regenerate themselves. Why is that not true for CAR T BCMA?
Why are they not regenerating themselves?
Correct.
Is that the question?
Yes.
Yeah, I mean, the question is, how do T cells undergo renewal? Where do they start from? Are these terminally differentiated T cells that are not able to undergo cell division and multiply? Will the gene that has been inserted get into the daughter cells? I think it's not a very simple answer. I think we're still learning what these cells are, which cells are we truly transferring to them, and are the progeny going to include the transfer gene? So stay tuned. I mean, these are only like 12 patients, so I think we need more patients and more analysis to understand this.
Great, thank you very much. And so that's going to conclude our webinar. I want to say thank you so much to Dr. Abonour for spending his time this morning answering our questions and going through his presentation. For those of you that are on that are GMAN members, Dr. Abonour will be at our summit this June. He has a short presentation and he will be able to answer some questions. So if you have any lingering questions, You can save it for our meeting in Copenhagen, Denmark. I also just want to take a brief moment to thank our sponsors, because without them, this would not be possible. So thank you so much to Celgene, Novartis, Bristol Myers Squibb, Amgen, and Takeda. And so thank you again, Dr. Abonour, and thank you everyone for listening. And everyone have a great morning or afternoon. Thank you, Dr. Abonour.
Thank you. Have a good day. Bye-bye.
All right. Bye-bye.